All right, welcome back to our discussion of Mendelian genetics. We're going to move into concept 14.3. So, inheritance patterns are often more complex than predicted by simple Mendelian genetics. So, um, when we look at Mendel's experiments, what I first notice is that he was extraordinarily lucky. So he just happened to pick the right pea plant characteristics to study. It rarely actually occurs this simply. Um, if he had picked something different, he may have really muddied the waters and not been able to come up with a single conclusion. The reason for this is inheritable characters are not always determined by only one gene with two alleles. We can have various alterations of this, characteristics that are involving more than one gene, or maybe you actually have more than two alleles. So it can get much more complex. Now, despite this, the basic principles of segregation and independent assortment still apply to these complex inheritance patterns. So we're going to begin to look at some of those. Now, we can get deviations from Mendelian patterns when we move into these following scenarios. So when alleles are not completely dominant or recessive, when a gene has more than two alleles, or when a gene can produce multiple phenotypes. So here, we presented a scenario before where something was completely dominant or recessive. Well, there's an in-between too, and there's codominance. There's a lot of variations here. As I said before, you can have more than two alleles. With multiple phenotypes, sometimes the phenotype is dependent upon where that gene is expressed. So in cystic fibrosis, you get a particular phenotype occurring in the lungs, and you get a separate phenotype occurring in the guts. You have a separate phenotype occurring on the skin. So it expresses itself differently depending on where that gene is at and what its overall function is. So before we looked at the idea of complete dominance, and we were looking at purple and white flowers, and if you had the enzyme that could produce the purple pigment, you had a purple flower. And if you didn't have that enzyme, you would have a white flower. If you had one of each, in the case of the heterozygote, you would still have a purple flower because you still had the enzyme that made the purple pigment. And that was enough to make the flower purple. Let's look at a scenario here that we call incomplete dominance. Okay, so here we again have a flower, and the scenario is largely the same. We have an enzyme which is producing a red pigment, and in the white version of the flower, we have a non-functional enzyme. So we create no red pigment. In this case, when we produce the F1 generation, we actually produce a pink flower. And that is because when we have only one copy of the functional enzyme, it is making exactly half the amount is if you had two functional copies. So here you would be, or up here, you would be creating two functional copies of that enzyme, and you'd make a certain level of product. Here we only have half the amount, so consequently we don't end up with a red flower, we end up with a pink flower. In the case with the purple and white flower in pea plants, it just so happened that one copy was enough to make the entire flower purple. It very easily could have been a different scenario. It's just how it worked out there. Now, in this case, when we go to the F2 generation, you can actually see that the phenotype and the genotype are similar in that it follows a 1 to 2 to 1 ratio. Remember, in the purple and pink flower and peas, where we had complete dominance, the phenotype was 3 to 1, and the genotype was 1 to 2 to 1. So here you can see an example of how that plays out. Now, in addition to incomplete dominance, which we just showed you an example of, we also have something called codominance. This is something that you may have already experienced. This is true in the case of blood types. So you know that you can have type A blood and you can have type B blood, but you can also have type AB blood. And as you can predict, that is true because both of those alleles are codominant. They are both expressed, and it leads to a different phenotype when we have both of them expressed. So I'll come back to that idea of blood because there's another factor in play there. But let's talk about a few other things first. So let's keep in mind that a dominant allele does not subdue a recessive allele, right? Both are still there, and those alleles don't really interact. So when we say an allele, we're simply talking about there's a variation in the gene nucleotide sequence that is going to lead to a change in the amino acid sequence of some type of protein. And when that happens, both of those proteins are made, whether it's functional or non-functional or has altered activities. So a dominant allele doesn't shut down the recessive. They are both still expressed. It just becomes a matter of, is one copy enough to make it work? Now, for any of these characteristics, this idea of dominance and recessiveness can really depend on what level we examine the phenotype. So let's take an example of something called Tay-Sachs disease. This is a fatal recessive trait, and it is caused by a dysfunctional enzyme which is involved in the breakdown of lipids. So consequently, in the disease state, you have an accumulation of these lipids in the brain, which causes a fatal neurological disorder. So here, when you look at the entire organism, that allele is recessive. If you have one functional copy of the enzyme, you do not get Tay-Sachs disease. However, if you take a look at the heterozygote and you look down at the biochemical level, you will recognize that the actual phenotype, which in this case is the enzyme activity level, not whether or not you have a disease or do not have a disease. Here at the biochemical level, we're simply talking about enzyme activity level. So here what we have is incomplete dominance. So we have a scenario where we have a non-functional version of the enzyme. It is still there. We have a functional version, and it is breaking down lipids, but it is not complete. When you look at a molecular level, thinking about a lipid interacting with a enzyme, whether it is the functional copy or the dysfunctional copy, the alleles are essentially codominant. If the lipid hits the functional enzyme, it'll get broken down. If it hits the dysfunctional enzyme, it'll not get broken down, right? So it's completely codominant at a molecular level. So sometimes it depends on how we're looking at it to distinguish what dominant and recessive actually means. Now, something else to consider is dominant alleles are not necessarily more common in populations than recessive alleles. So an example of this is one baby out of 400 will be born with extra fingers or toes. The gene that regulates this is actually dominant, right? So it's not only it just simply comes down to dominance is not necessarily related to how common that allele is.